afternoon, everyone. Okay, good afternoon. And as promised last week, during my class, I promised that last week I did a class on Ujian Amali Science, which is coming soon, actually next week. So I promised last week that I'm going to have another lesson, which is today, on how to answer defining something operationally. Or what in BM we say mendefinisi secara operasi. Okay, because this is one of the very more difficult questions to tackle. All right, so a lot of students always ask me, teacher, how do we answer this defining operationally? So the thing about this question is, you don't have one fixed answer, even though you have experiment A talking about diffusion, all right? And you'll be using different things to experiment for diffusion. And then you have experiment B, and so talking about diffusion or experimenting about diffusion. Your answer for defining operationally will not be the same for these two experiments. Okay, so today, I hope that after going through my class today, that all of you will be able to get an idea how to answer defining operationally and get the maximum mark. Okay, sometimes it will be 2 marks, sometimes it will be 3 marks. Okay, all right, so nice to see you, Huzaifa, right? And Madhu Mehta Selva Kumar. Okay, good afternoon and welcome to my lesson. Okay, so I'm going to share screen now. All right, I think you can see here already. So let me change the layout. All right, okay, so you see less of me and you see more of the screen. All right, okay, so let me go through. Okay, Huang Jingle. Okay, hello. Yeah, nice to see you and thank you for coming to my class to learn. All right, hopefully all of you are able to learn, and by the end of today's lesson, you will have a very clear idea how to answer for this defined operation. Okay, all right. So now let's start. Let's get started. Okay, so this is my thumbnail for today's lesson. All right. Now, first of all, now this is the 9th construct. If you have not seen my last week's video, I explained about 11 constructs. There are 11 types of questions that can be asked in your Ujian Amali Science. Okay, ada 11 soalan yang boleh ditanya lah. Alright, so my slides will be in double language, Dui language, that is BM in English. So if you don't quite understand or you're not learning in English, it's alright. My answers all are there in BM as well. Okay, so the thing to remember for defining operationally is there are 3 main ideas. In your answer for defining operationally, you should include these 3 things. Okay, so 3 perkara that you have to put inside your answer. Okay, first thing here is, all right, what is X? Let's say, for example, what is diffusion? Define operationally diffusion. Okay, let's say experiments about diffusion, right? Molecules moving in and out, depending on what molecule, you have to see the question. That's why this question, defining operationally, there's no fixed answer. You have to see what is used in your experiment, and it must be specific. All right, you cannot give a general answer. Diffusion is the process of molecules moving from a region of higher concentration to a region of lower concentration. That is a general definition, which is not wrong actually, but it does not answer this question. Okay, it is not what they want. So if you answer like that, all right, you may not get your marks because it is not wrong. It is the correct definition for diffusion, but it is wrong in the sense that you did not answer defining operationally. It is a definition, but it's not operational definition. Okay, so operational definition, please remember that it must be specific based on this experiment only. This definition that you write for this experiment will not be able to be used for another experiment that also talk about diffusion. Understand? All right. Okay. Now, later when I give you contoh soalan, all right, the examples of questions, you will get a clearer idea how to formulate your defining operationally. Okay. So the second idea here is how is your X, that means your diffusion, let's say diffusion or maybe photosynthesis or transpiration, how is it measured in this experiment? Not generally how to measure transpiration, how to measure photosynthesis, but in this experiment, what is used to show the rate of photosynthesis. Maybe the number of bubbles that are released in 5 minutes. That shows how fast is your photosynthesis. Okay, so what is the rate of water loss when you carry out transpiration? You know, water will evaporate, isn't it, from your leaves, from the leaf of the plant. So how fast is the water disappearing? That is, you must mention for the second point here. Okay, and the third point you have to mention is what affects your this X. X means like either diffusion or whatever they ask, like photosynthesis or transpiration. What affects it? What is your MV, in other words? Okay, so the first point is what is X? If a statement, all right. Later you see lah, huh? And then second point is how is it measured? That means you have to mention your RV, responding variable. Pemboleh ubah yang bergerak balas. Okay. And the third point is what is your MV that affects the thing you're talking about. So you have to mention the manipulated variable. Okay, so if you mention all these three facts, all those three ideas in your defining operationally, you are very safe. Okay, now let's go through questions. Like I said, we don't have a fast fixed rule. This question must say this. You have to see what was used in your experiment. Okay, that's why I have to give you contour soalan. If I don't give you the question, I don't give you the apparatus, you will not be able to catch it. Okay, so I give you experiment example. Now I've got 10 questions here. Okay, 10 questions here, experiment questions, which were taken from last time. Last time they had paper 3, yeah. Okay, now like last time, you know, before your KSSM syllabus, we have a KBSM syllabus, whereby the students also carry out an experiment, but it's not practical. We don't have Ujian Amali Science before 2021. Okay, before 2021, that means 2020 and before that, they have a paper 3. The paper 3 is actually a written experiment. That means they don't have the apparatus, they don't need to do the practical. Everything is printed on the paper with the readings, with the diagram. They just have to write the answer on paper. Okay, so that was before 2021. 2021 is the first year of Ujian Amali Science. So your year now is the 5th year. Okay, the 5th year, 2025, is the 5th time they are having Ujian Amali Science. Okay, now let's look at the question. I'm sure you can recognize on the diagram, you know what experiment it is. It is an experiment to carry out to determine the energy value of 3 types of food. Okay, so later you will see in the procedure or whatever, or the list of apparatus and materials, what are the 3 types of food given to you, right? When food is burnt, energy is released. Okay, that way you should know that from theory, because chemical energy is stored in the food. Okay, when you burn it, it is releasing. Okay, heat is released, and that heat is transferred to the water, the distilled water. Distilled water absorbs the heat and the temperature rises. You take the reading of the rise in temperature, okay, perubahan the kenaikan suhu, and then you put everything, all the values into a formula, a fixed formula, yeah, right? And you can find the calorie, or we call the energy value. Okay, so now you have to see what is given to you. So let's see the table of results first. So the table of results, they give you the sample, all right? If you do not, of course, this one is before your experiment, it means before your Ujian Amali Science 2021. So this is all given to you. But if you are doing the experiment yourself, you have to draw your own table. Okay, draw a table of result. So you draw your own table. Okay, now this is your MV. You should know that in any table, you must have MV. What did you experiment? What did you burn? You burn the 3 types of food. That is your food sample. That is your manipulated variable. Okay, and what did you measure? You measure the final temperature of distilled water, the rise of temperature in the water. This is your RV. Okay, now take note of your MV, RV. Both of these must be in your answer for defining operationally. Okay, so based on experiment, on this experiment, this is the question. Define operationally the energy value of food. Okay, so the first point is you must start your answer with saying energy value is. You are answering what is the thing they ask. They ask you what is energy value of food. So you start your sentence with energy value of food is what? Okay, is what? So is the heat or the energy that is released by food sample when it is completely burned. This one is from theory. If you don't burn it completely, some of the energy is still locked inside the food molecule. You did not release it. So your reading will not be, your data will not be accurate. Okay, so you must mention the first point is energy value is the heat or is the energy released by food sample when it is completely burned. Okay, right. Now, second point. Remember the second point? You talk about the RV. What was used to show this energy value? Okay, you see that we measured the final temperature of distilled water, which is shown by an increase in the temperature of distilled water. That means you take the initial, then you take the final. After burning is the final, you'll see the difference there. That is the increase in temperature. So this is what you measure. So your P2, that means your second point, you mentioned about the RV. And the third point, you mentioned about what affects your energy value. And then you see carefully. So this is your second point here, from what you measure, shown by what, determined by what. Okay. Next, manipulated variable, you write on the third point. That means energy value is affected or influenced by the type or the class of food. Okay, so you see here, P1 is defining it, what is energy value. P2, what do you use to measure your energy value, your RV. Third point, what can affect your energy value? Your NV. Okay, right, clear? So this was a past year question. Okay, but not your year because this was from before the 2021. Okay, now we go to the second question example. All right, any questions, just type here and I'll try to answer it. All right, okay. Second experiment. So look at what you're given. All right, you are given amylase solution, you have starch here, so it's hydrolysis of starch by amylase. Okay, you put into a water bath, which is at certain temperature, which is the 37 degrees Celsius. 37 degrees Celsius is the optimal temperature for activity of enzyme. So usually we will put warm water, right? 37. Okay, question. An experiment was carried out to study the effect of pH value. So your pH value is the buffer solution. That means if you want to have pH 7, you will add in distilled water. If you want your pH to be acidic, you will add acid, hydrochloric acid usually. If you want to test it, you want to test this alkaline condition, you will add in alkaline. Okay, so that is your buffer solution, which has a different pH. All right. Yeah, Huang Jing, Muhammad Farish, yes. Good evening, good afternoon. Yeah. All right. Nice of you to join me. Okay, so now let's say this is the result. Now, like I said, all this is already given because this is a written experiment. We call it written experiment. So you want to see different pH. So that means there must be at least three values. So here they give you 1, 2, 3, 4, 5 values. Okay, and how do you test hydrolysis of starch? How do you know the starch has been hydrolyzed or not? Anybody know? Okay, there's a question here. Do we need to connect the x-intercept? Oh, x-intercept means you extrapolate, isn't it? Jinglet. Extrapolate means you draw the graph backwards, right? Draw the graph backwards. For biology, don't. Biology, no. Leave your first point, your first dot, and the last dot. You join it. If it's curved, you need to draw a curved line. Smooth curve line. Don't have all the bulu-bulu, you know, you draw like you're doing your surname. No, you just one nice straight line and connect it. If it is very obvious that all your points fall on the same line, that means it's a straight line, you need to use ruler. Okay, so depends on your graph, and please don't extend your graph backwards. Don't extend your line forwards. We call it extrapolate. You don't do that for bio. Yeah, okay, KJX, correct. No need for bio. You need, if you want to confirm with your whether you can do that for physics or biology, you better ask your biology or whether you can do it for chemistry or physics, please ask your chemistry or physics teacher. But for biology, confirm. You do not extrapolate. Just start from the first X, the dot, connect to the last dot, leave it like that. Okay, right. Yeah, okay, welcome, Jingler. I hope it answers the question. Okay, now we go back to this one. Hydrolysis of starch. There's only one thing you test with starch. Anybody knows? What do you test starch with? How do you know there's presence of starch? Anybody? Jingler, Farish, who else? KJX, and Selva Kumar, Madhumata. Okay. How do we know that whether the starch has been hydrolyzed or not? Okay, simple. There's only one substance you need to use to test for presence of starch. Okay, for the answer? No answer. All right. Maybe some of you very shy. You should know, right? I think you know. Most of you will know already. Just that you're very shy. I don't want to make yourself known. I see there's more than 10 people here. 20 maybe. All right, more than 20. Yes, good, Elsa. Iodine test. Yeah, you need to put iodine. Your iodine is a reagent. When it comes into contact with starch, it will turn blue-black or dark blue. Okay, so look at this experiment. So when you do the experiment, you will have all these dots, or we call blobs of iodine all prepared already. You just take one drop of your solution. If you drop it there and it turns dark blue, that means starch is still present. Okay, now let's say, this is the MV. Always look at a table. There should be MV, there should be RV, what you measure. Okay, no CV, no constant variable in your diagram, in your table. Okay, now what is the question? Based on this experiment, define operationally hydrolysis of starch. Okay, so what is hydrolysis of starch based on experiment? You see, you have albumin, you have amylase. So when the albumin, that is the starch. Starch and amylase, yeah, correct. Starch and amylase. Starch will be hydrolyzed by amylase, all right? And then once it's hydrolyzed, it's no longer starch. There's no starch. When you drop it onto the iodine should not change dark blue. The iodine will be the original color, which is yellow. Okay, so now, first point, hydrolysis of starch is, you must start is, okay? Hydrolysis of starch is the action of amylase on breaking down starch. You see, you use amylase in your experiment. You also have starch there. This is your substrate. So hydrolysis of starch is the action of amylase on breaking down starch. And see what is the RV. How do you know whether the hydrolysis has completed? You look at the RV, which is shown by the time taken for iodine solution to remain yellow. You see, we just look at it straight away from the table, the RV. And your third point, what's the third point? The third point is hydrolysis of starch, or say it is affected by MV. What is MV? MV is the pH value of the solution. Okay, see, you can catch or not? You can catch on first point, you must say, what is it? X is something, something, something. Second point, it is shown by the RV. Third point, it is affected by the MV. Okay, clear? Right. 2 experiments. Let's go on. We've got 8 more to go. Right now, this one. Question 3. This one shows you take sample of water sample from different location, and you want to know whether it's polluted or not. Okay, this one from chapter chapter 9, chapter 8, chapter 8, from chapter 9, yeah, chapter 9 in your form 5, okay, the ecology, right? So diagram 3 shows the apparatus set up to study the level of water pollution in 4 different water samples. So you got P, Q, R, and S. Maybe take from the pond, take from the longgang, take from the rainwater, or take from the river, okay, let's say. Okay, they are tested using methylene blue. Okay, so methylene blue is actually dark blue. You add a little bit of methylene blue, that's about 1 cm or 1 ml, into a sample of water, and then you close it up and you put it in a dark place. And then what you need to know, what you need to measure is how long does it take for this water, because when you put methylene blue, it becomes blue color. How long does it take for your water that is colored blue to become colorless? Okay, once it changes to become colorless, it means the oxygen is no longer there. There's no oxygen. That means the faster it changes color, the more polluted your water is. Remember that. Okay, so this is to measure the BOD level. So when the stopwatch, you need to use stopwatch. If the time is very short, very fast, the color changes from blue to colorless, it means that water is polluted. Okay, now let's see the reading. Okay, let's say for location P, location Q, and then the others I didn't put, like R and S, I didn't put because too long. So you don't need that. I just want to explain this. It doesn't need all that. So MV, your sample water, because you got 4 samples. RV, what do you measure? Time taken for the decolorization of methylene blue solution. That means from blue to colorless. Okay, this is your RV. Okay, question. Based on the experiment, define operationally polluted water. So what is polluted water? Based on what you did for this experiment. Okay, so it has to be specific. So specific means you must unveil what you use and put inside your definition. Okay, so answer. First point, polluted water is water sample taken from different locations, which is P, Q, R, and S. You use 4 different samples of water. Okay, these are the 4 different locations. And how do you know it's polluted? How polluted it is? It is shown by, determined by the time taken to decolorize, or just copy there straight away, for decolorization of methylene blue solution. Okay, and your third point, pollution, water pollution, okay, or polluted water, right, is affected by the source of water sample, which is your NV. Okay, so X is affected by NV. This is your basic formula for the third point. Your X is affected by NV. Okay, you can catch it now. The third one is always easy. The third point is the easiest one. Okay, all right. Okay, no questions so far. Right, I go on to question 4. Yeah? Okay, so you see this is to investigate transpiration. All right, transpiration is affected by a few factors. So here we are talking about air humidity. So you can see, read carefully. In the question, they will always give you what factor they are going to investigate. Usually they're investigating air humidity. So they want to investigate whether the air is dry or the air is wet. How does it affect the evaporation of water from the leaves, which we call transpiration. Okay, so your air humidity is your MV. Your transpiration or rate of transpiration is your RV. Okay, so this experiment is carried out for 30 minutes. So how do you change the humidity? Okay, simple. Look at the 2 diagrams. What is different? What is different? Can you see that? What is different? Everything the same, right? Except for one thing, which is, you see the one on the left-hand side, you don't have this anhydrous calcium chloride. Anhydrous means kering. Kering means dry, right? Calcium chloride, anhydrous calcium chloride, is used to absorb moisture in the air. So if you have a plastic bag over your plant, the one on the left-hand side, no anhydrous calcium chloride. That means the air here is got water That means it's humid That is water, not absorbed by the calcium chloride. Okay, but the one on the right-hand side, because you have it there, the function of calcium chloride, anhydrous calcium chloride, is to absorb moisture. Okay, so I have a question here. Provide CO2. Oh, no, Atira, the provide CO2 is not this. You must have confused between this one, calcium chloride, kontang, right? And the other one, which provides CO2, is called sodium bicarbonate solution or sodium hydrogen carbonate. Okay, I'll type it here, sodium hydrogen carbonate. That one, actually, you put in the water for the aquatic plant when you do your photosynthesis experiment to see whether how fast the bubbles come up. That one is to provide oxygen, carbon dioxide. Okay, so no need for me to type. Okay, no need for me to type. It's sodium bicarbonate or sodium hydrogen carbonate. It's the same. Okay, so remember this is to absorb moisture. So you see the difference here? Your air humidity is affected by the presence. Okay, not presence because different amounts. Later you'll see got 0, got 10 grams, 5 grams, whatever. So this is the function is to change the air humidity. Okay. Now, so here you got 0 gram. That means you don't have calcium chloride. All right. And the another one is 50 grams. You put 50 grams in the plastic bag there. You need to measure how much water, what is the mass of the water. And at the end of it, after 30 minutes, you measure again your apparatus to see how much water has evaporated. That means loss of water. Okay. The more water loss, that means in a shorter time, that will be the rate of transpiration. Okay, question. Now this is your MV. Okay. And that is your RV, the mass of water, the change in the mass, you must minus from the original, from the initial. Yeah. Okay, now see the question. Question here is based on the experiment. Always when they ask defining operationally, they will always say based on this experiment or based on the experiment, because your answer is only for this experiment. You cannot use this answer in another experiment for translation. Okay, now what answer? Okay, you should start with transpiration is. Okay, always remember, you start with a definition. You want to explain something, you must say this is. So transpiration is the loss of water from the balsam plant. This one have to be specific, because in your experiment, if you look at the previous diagram, okay, go back. You see the diagram here? What plant did they use? They use balsam plant. So if they use balsam plant, your answer here must be specific. You cannot say plant, general, cannot. You must say balsam plant. If you say the experiment referring to hydrilla in the water and all that, you must say hydrilla species. Okay, so you must write the specimen that was used. Okay, clear? Right, so we go back to this one. Loss of water from the balsam plant. No balsam plant, no word balsam. Don't have one. Okay, and what is shown by what? Transpiration is shown by what? Shown by the RV, the change in mass. Because initial and final means the difference, right? Or the difference in mass of photometer in 30 minutes. We did the experiment for 30 minutes. So specific for this experiment only. Okay, and the third point everybody can answer, right? It is affected, it is influenced by mass, the mass of anhydrous calcium chloride means air humidity. Okay, because your anhydrous calcium chloride is to absorb water. So when it absorbs water, it actually controls the air humidity. Okay, you can also write affected by the mass of anhydrous calcium chloride. Not wrong. You can also say that. Okay, it's not wrong. Okay, go to fifth question. Now this one you can see is ascorbic acid. All right, testing for vitamin C using DCPIP. You need DCPIP, which is a solution. All right, that is going to be, when you add your vitamin C, the color of your DCPIP will become colorless. It will change color. Okay, and you must add in enough of your solution your vitamin C or whatever, until the color of this BRP becomes totally colorless. Then you stop. Then you see how much solution you need. Okay, go and revise this if you're not sure. Okay, diagram diagram 5 shows the apparatus set up to determine the concentration of vitamin C in fruit juices using BCPIP solution. So you must start with the standard solution first. Standard solution is they prepare for you 0.1%, usually 0.1% of ascorbic acid. Actually, ascorbic acid is vitamin C. Vitamin C is actually ascorbic acid. Okay, where can you get this module? Okay, I don't have a module, but actually, if you want to put passive questions, you can get from the all these what you call the book you know, some books they will publish the passive question. So what I'm doing here is all passive questions. Passive question, actual SPM question. Okay, so I don't have a module, but what you can do is I just copy questions from here and there, and I just prepared the slide lah. So I don't have a module, but what I normally prepare is just all these videos. You can have a look at the videos. Okay. In the module, I think you have other groups, bio groups in the Telegram. You join there, you can get a lot of material. Okay. All right. Okay. Now, so what is the question? Question here. Wait, before that, MV. This is your MV. This is your RV. All right. As usual. You must put in the solution, right? You first test with ascorbic acid first to get your standard. How much you need to put inside? Okay, how much you normally put 5 ml next in the syringe. Then when you put in, you press your the piston of your syringe. The solution goes in. Then you of course you need to stir as you are going along. Stir this blanca berjaga-jaga. You stir and make sure do not press too much. You have to stir slowly and see the color change. Once it has decolorized, you stop adding already. You stop. And then you read how much solution you need. 5 ml minus, let's say the balance is 3. So 5 minus 3, that means 2 has gone inside. 2 ml has gone inside your UCPL. Okay, so you fill in the table. This is the MV, this is the RV. Now, defining operationally. What is vitamin C? Okay, define operationally vitamin C. So vitamin C is ascorbic acid. It is ascorbic acid. Okay, ascorbic acid, but not just any ascorbic acid. Like I mentioned, it's specific for the question. The question here, we use what? Papaya juice, guava juice, mango juice. So you write all these. Okay, what did you use? Like for example, just now the balsam plant. You must write the word balsam plant inside there. When you are given all these 3 juices, you have to write inside this 3 Okay. Second point. The second point, I think you are getting there already. You more or less know, which is shown by what? The volume RV lah. Shown by the the volume of juice or volume of solution to be colorized. How much? Because what? 1 ml. You must be specific. You cannot just say this colorized BCPIP. You must say 1 ml of 0.1% BCPIP solution. Just copy anyone, okay? Because it's given in the question already. Okay, and the third point, easiest one, the concentration of vitamin C is affected by the type of fruit juice. Okay, so maybe your papaya do have less vitamin C. Your guava has more. Your mango will have somewhere in between. So it is dependent on the type of juice. So different juices will have different concentration of vitamin C. Okay, right? Okay, Arifa, okay, so you third point, not really inference, Arifa. If inference is actually to explain your observation. That's the meaning of inference. Okay. I think we are talking about hypothesis, is it? More like hypothesis. Okay, that's more like inference is actually, if you see my last week's lesson, last Saturday, last week, I gave a general idea, inference, observation, what is needed. So inference is actually a reason. If you are giving the reason for your observation, why do you get this result? Because blah, 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 blah. That's inference. Inference is the reason for your observation. Explaining why you get this observation. Okay, so I think what you mean is like hypothesis. Okay, Right, next one. Question 6. Okay, now so you have your whisking tubing here. All right, now this is not so simple because you have the capillary tube connected into the whisking tubing. All right, and then your whisking tubing is filled with water, distilled water. Okay, outside is sucrose solution. Sucrose solution. That means outside now is hypertonic. Okay, compared to the inside your whisking tubing is hypotonic. Okay, and you know this is osmosis happening. You'll find that outside is sucrose, inside is distilled water. Water will actually how? Diffuse from a region of theory from a region of high water potential to a region of low water potential. So high water means it's inside. Inside is just pure water. So you got a lot of water inside there. Outside is less water because you got more of sucrose. Okay, so from what happened is you got the thing water should move out. Water should move out. Okay, so when water moves out, the capillarity, the water, the liquid inside there will move as well. Okay, so you see, right? Wait, question first. Diagram 6 shows an experiment to study the rate of movement of substances across a whisking tubing. The whisking tubing containing distilled water is immersed in different concentrations of sucrose solution for 20 minutes. Okay, so the thing to see is you got to, think you got to use your gray cells a little bit So when you do the experiment, you need to know that osmosis will occur, and all right, the X here is the original, what you call the initial reading Your water is until this level. Then when osmosis occurs, water is in the middle, right? Water is inside this hypotonic. Water will diffuse out of the whisking tubing. Therefore, the level will drop, right? The water inside will become less. So this capillary tube, the water there will move inwards. Okay, so like this when water goes out, the water will come in right up. So this will move. So now what you need to measure is the how many cm. Okay, to show the what they call the how much water has moved out from the whisking tubing. Okay, understand? So now let's say this is the reading. Okay, like I told you again, this is from past year. They actually give you some readings already on paper. You just need to answer on you don't need to do the experiment. Okay, but if you were to do the experiment, you got to draw your own table, and you measure yourself the distance, how many cm. Okay, so what's the question? MV first. This is your MV because you are doing different concentration. You do first time, second time, third time, fourth time, everything the same. You only change the concentration of the sucrose. What you measure? You measure the distance, how much water has moved in. Okay, the more concentrated the sucrose is, more water will leave the whisking tubing. Therefore, this distance will become longer. Okay, more water will go inside. Okay, so what's the question? The question here is based on experiment, always, right? They always write in front there. Define operationally diffusion. So your diffusion is specific for this question, where you have your capillary tube your X and your Y what is the sucrose solution this and all that. You cannot apply this answer anywhere else. Okay, so first point, diffusion is movement of water. You cannot say diffusion is time. No, not time. Diffusion is distance. No, diffusion not distance. Diffusion is the movement, of water molecules out of whisking tubing into the sucrose solution. So you got to be specific. What is used? You have your water, you have sucrose there, and the movement is from inside the whisking tubing outside. Okay, so this is specific to what you were given. Okay, next. Second point. It is shown by what? It is shown by what? Shown by the distance from X to Y. You see, the more concentrated the sucrose solution is, the more water will be going into the whisking tubing to replace the water that is lost, the water that came out. Okay, the water will go inside. All right, and then the third point, it is affected. Diffusion is affected by MV. Influence affected similarly. Concentration of people. Okay, more or less you know already, right? Now getting, you know, as more questions I give you, you have a steadier idea, right? Okay, we're going to question 7. Okay, now this one is to determine how much carbon dioxide is in your exhaled air. Let's say you are given experiment now. Of course, quite unlikely you won't be doing it for your ujian amanasan because it's very time consuming, right? The person has to do activity, exercise. So that's why this is for theory, you know, the experiment is theory one. Okay, that means you write down the values. Okay, an experiment is carried out to study the effect of different duration activity. So a person doing different exercise, okay, different time, not exercise, same exercise, different time only. Running on the spot for 1 minute, 2 minutes, 3 minutes, exercise is the same, running on the spot, okay? And then after the person has done it, so take a sample of the exhaled air from the person. And then this exhaled air is absorbed into this, just take a sample of it using a J tube. After that, you analyze the amount of CO2 in the J tube by using something that absorbs your CO2. This one you say Arifa, that one correct, okay? You can also use potassium hydroxide to absorb. That one is to supply CO2, right? Just now your answer is to provide CO2 is sodium hydrogen carbonate. This one is to absorb CO2. Absorb CO2 is using potassium hydroxide. Okay, see carefully, Now, so your MV is the time. Okay, actually 0, 1, 2, 3, the time taken to do the activity, the time taken. Okay, not activity, activity is the same, just that the time taken. And how do you know how much carbon dioxide is inside? You got to take a substance to absorb the carbon dioxide, and then the air column will become shorter. When the CO2 has gone into the solution, the air column will become shorter. Then you take the distance, take the length of your air column, divide by the original times 100%. Then you will get your percentage of carbon dioxide. Okay, so that's why using potassium hydroxide is to absorb CO2. Okay, Arifa, absorb CO2, you use potassium hydroxide. Provide CO2 is using sodium hydrogen carbonate. Okay, and just now that one is using anhydrous calcium chloride is to absorb water or call it air molecules in the air. Okay, water moisture. Okay, now what's the question? Question here is define operationally exhaled air. So what is exhaled air? Okay, what is exhaled air? So exhaled air is, again, always answer. First question, first sentence, exhaled air, what is it they ask? Is. Okay, is what? Is the air sample that contains carbon dioxide. Okay, it must contain carbon dioxide, otherwise you're testing carbon dioxide here now. How much carbon dioxide is in the exhaled air after you do different time for the activity. Okay, you run 1 minute, run 2 minutes, 1 3 minutes, you get carbon dioxide in the air. Okay, and number 2, second point, how do you get the amount of carbon dioxide? It will be absorbed by the potassium hydroxide solution in the J tube. You use the J tube to measure that, to find out. So remember, whatever was used, go and campak inside, go and sumbat inside your answer. Then P3, it is affected. Okay, the length of the air column is affected by the duration of the activity. That means the time. See, 0, 1, 2, 3. That is the time used to carry out the activity. Not type of activity. You see, because running on spot is the same. That is not activity, the type of activity, rather the muscle, tempo, or the duration. Okay, question number 8. Okay, this is to experiment to investigate photosynthesis. Now this one, see this, Arifa, sodium hydrogen carbonate. Okay, or we call it BM, natrium hydrogen carbonate in BM lah. So this is to provide your carbon dioxide for the photosynthesis. Okay, now what's the question? An experiment was carried out to study the effect of light intensity. You can see the bulb there. Okay, and it's affected by the distance. What we do is the ruler here to show the distance. The further you put your light, the light intensity is lower. The nearer you put your sunlight, the bulb, the light intensity is higher. Okay, all right. So now see, you use what? Hydrilla. Hydrilla. So you make sure you must masukkan the name hydrilla in your answer. Okay, okay, good. Arifa, you get it, right? Okay, now so this is your answer. Let's say 60 cm, 50 cm. There are some more like 40, 30, 20. No need to write so many. It doesn't affect my answer. So MD is here. The distance, because this is what you change. 60, 50, 40. You do a few times. Okay. And what do you measure? You measure using a stopwatch, right? You measure 5 minutes. How many number of bubbles came out during the 5 minutes? Okay. So this is roughly what came out last year for Ujian Harmonic Science. Where they have the lampu, right? The menthol, right? The lamp. And then you also count the number of bubbles. That was last year's Ujian Harmonic Science. Okay, but not this question. This question came out earlier. This is the before 2021 one. Okay, also came out, but this is written as well. Okay, so let's see the question. Define operationally photosynthesis. So what is photosynthesis based on this experiment? Okay, so what is photosynthesis? So you must start first point. Photosynthesis is what? Is what? Is a process, right? Photosynthesis is a process. It is not a value. It's not something, right? It's not an object. It is a process occurring in Hydrilla species. So you write the name of this plant. It is a process that happens in Hydrilla species in sodium hydrogen carbonate solution. Okay, you provided that also in the experiment. And P2, shown by what? Your photosynthesis is shown by what? How to measure how fast or how slow is your photosynthesis. You see the number of bubbles, right? Not P2, which produces gas bubbles. Gas bubble. Whether you got photosynthesis, you see the bubbles because the bubble is oxygen. Oxygen is produced. So when there is bubble that comes out, you know that there is photosynthesis. And what about P3? Third point, the rates. How fast or how slow the bubbles are being released is affected by the distance between the light source and the hydrilla. Okay, affected by the distance from light source, or we call it light intensity. So light source, the distance is actually representing the light intensity. So the third point is always the MD. Okay, right. So question 9. Okay, coming to the end. So this one is albumin. This is the protein, and you add in pepsin. So you want to see this is hydrolysis of protein, hydrolysis of albumin. Just now the earlier one was starch. So this is hydrolysis of albumin using pepsin. And you can see in the beginning actually, albumin is large molecule. So it should be cro. Cro means it's cloudy. Cloudy means you cannot see through. It's blur, right? Blur. So it's very putih like that, very blur. You can't see through because the molecules are not soluble. Molecules that are big, they are usually not soluble. Okay, but once hydrolysis has occurred, the albumin will be broken into smaller units. Okay, maybe it becomes smaller units of polypeptide, peptides, and all that. So it's clear because it is easier to dissolve. Okay, so once you see this one is like no values to take, you see the clarity of your solution. Okay, if it becomes clear, then it's an indication that your albumin has been hydrolyzed. Okay, see question. An experiment was carried out to study the effect of concentration of albumin suspension. So you do a few different suspension, the concentration on the rate of reaction of pepsin enzyme called pepsin. Okay, pepsin enzyme. So the time taken for the change in cloudiness. So you see the time, whether it turns cloudy fast or it takes a long time. Whether it turns clear fast or it takes a long time to become clear. It shows you whether the rate of enzyme is high or low. Okay? If it takes a long time for it to become clear, that means your enzyme is not working very well. If the albumin becomes clear very fast, that means your enzyme is working very, very good, very well. The rate of enzyme reaction is high. Okay, so now you see the time taken. Okay, look at the result. Yeah, the first one used 10%. Okay, then the second one used 15%. Your volume is the same, right? And then see how much time is taken before it becomes clear. So the end of experiment means it's clear. So you see C should be clear. That means you can see through. All right. So this is MV. Percentage of concentration is MV. And your RV is the, sorry, I put here, time taken. Oh, sorry, not time taken. It is the clarity, right? How clear. Okay, so define operationally what is enzyme. What is enzyme? Okay. Enzyme is a substance that hydrolyzes albumin suspension, because this experiment you use albumin. You cannot say starch already. It's not applicable for another experiment, only for this one. Okay, so enzyme is something, a substance that can break down. You can say digest, you can say break down, you can say hydrolyze albumin suspension, because this is what you use. Okay, and what happens is, how do you know it has already hydrolyzed? Okay, you see that it became clear. Okay, it became clear to show that it has already been hydrolyzed, because when you break down the molecule into smaller units, it will be easier to dissolve, so you can see through. Okay, suspension, we call it albumin suspension because it's not dissolvable, it didn't dissolve. But once it dissolves, it should be a solution. So you see, your suspension and solution, you should not use interchangeable. Something that does not dissolve, you say suspension. For example, flour suspension, starch suspension. When you prepare your starch suspension, you see it's blur, right? It's kero. Kero means it's cloudy, right? It's not, you cannot see through. But if it's sugar or salt, you never say suspension. You say solution, because salt and sugar can dissolve in water. All right, okay, so to become clear or becomes not turbid, and what is the third one? Again, MV, the rate of enzyme reaction is affected by the concentration of albumin suspension. So that is your MV. Understand or not? Okay, so you see a few examples, you're not sure, see again later. Okay, write down, think, how do I write my P1, P2, and my P3 specific for the experiment. Okay, one more, last one. This one came out 2019. All right, so I have to give a little bit of introduction first. Now, this is to test the effect of different sizes of the agar. Agar is, agar-agar lah, right? They put a chemical inside there. Okay, the chemical inside there is actually, now this 4 agar is already put a chemical. The chemical is called phenolphthalein. So when you put the chemical inside the phenolphthalein, the whole thing becomes pink color, because you already put alkaline. Alkaline and phenolphthalein, you know your chemistry, right? Phenolphthalein plus alkaline, you get pink color. So the entire cube here is pink color. But once you put it in acid, the acid will diffuse inside. It will react with your alkaline. It becomes neutral. When it becomes neutral, the pink color of the phenolphthalein will disappear. Okay, so you want to see how fast the hydrochloric acid goes inside, or you call diffusion, diffuse inside. All right, you need to put it into the hydrochloric acid to measure the time taken, right? Okay, that's not to say measure time taken, but you want to see which different sizes. You find 4 different sizes. One will be the fastest to change color to become colorless. The other one will be the slowest. Actually, what are you testing? You are testing the total surface area per volume measure. We call it TSA per V. Okay, the bigger the cube, the bigger the cube, the TSA per V is actually small. The smaller the cube, the TSA per V is actually bigger. Okay, you don't believe me? You go and find the total surface area of a small cube. You divide by the volume, get your answer. You compare with a bigger volume, go and find the total surface area. Then you divide by the volume, you compare your two answers, you will find that the bigger cube will have a smaller TSA per bit, while a smaller cube will have a bigger TSA per bit. Okay, so this is important for you also, for your paper 2 also. And you need to know the theory behind it. Okay, so now you immerse it, all right? And then now you see the time taken. See how much time does it take for your entire cube to become colorless. When it becomes colorless, it means your acid has gone in all the way, reacted with your alkaline, neutralize it, and your phenolphthalein turns clear. Originally, it's pink color. So once your acid goes in, reacts with your alkaline, becomes neutral, the the pink color disappears. Okay? Now, and then here, your MV is actually your PSA per V. So your small cube, J, you see, 1.2, bigger number. The M is the biggest cube. Okay, don't believe me, show me. See, J is the smallest cube. M is the biggest cube. If you do the calculation, you take your TSA divided by the V, you find that your J has the bigger answer, bigger value. Your big cube, which is the M, will have a smaller TSA per V. Okay, and then time taken depends on your experiment now, right? This one I'm going to write for you. So what is the RV is your time taken now? How much time you have to wait before the thing becomes completely colorless? Okay, so question. State the operational definition for rate of diffusion. Now, you see, just now the diffusion here, you cannot use the whisking tubing already, right? It's also diffusion, right? Not that one is whisking tubing, sucrose lah, water lah, capillary tube lah. You see, you're defining operationally, they cannot be used for all experiment. So here you have to write this. Okay, first, what is rate of diffusion? Rate of diffusion is the process. It's a process because diffusion is a process. But because you say rate is per time. So you have to write per time. Yeah, okay. So the process of movement of hydrochloric acid. So specific, not just water. It's not water moving. It is hydrochloric acid moving into the agar per time. Why you say per time? Because the question asks for rate. See now, you see here? Rate is always per time. If they don't ask for rate, you say diffusion. If they ask you what is diffusion, rate define operationally diffusion. Your answer will be diffusion is the process of movement of hydrochloric acid into agar cube. No need per time. Because rate, you must say per time. Okay, second point. Second point is what? Your RV lah. Okay, how do you know how fast is the rate of diffusion? Shown by what? Determined by what? Your RV. The time taken for agar tube to become colorless. You see that, right? And the third point is what affects your rate of diffusion? Look at your MV, is your total surface area per home. Okay, so not so correct to say size of the cube, but what actually affects is your TSA per bit. In BM, we say jumlah luas permukaan per isi padat, JLP per I. Okay, understand not? All right. Okay, Rehan, just join me now, is it? Adam. All right, Adam, Rehan. So you can watch the recording later lah. Go back and see. The link is always there, and my video is always there. You haven't missed out anything, go through again. See, have a look. Okay. So now I've given you 10 questions. Okay, go and analyze it and see. What I need to write for P1, P2. Remember the basic rule here is you have, if the plant or the specimen is given, you have to put inside. Okay, you have to put inside that. Second point is RV. How do you, is determined by what? Shown by what? The RV. The third point, your X or whatever you're answering is is affected by the MV. Okay, so depends on what MV you have lah. All right, so this one is very specific for the question one. So I can't tell you what is going to come out because I don't know. And also, even if I know, let's say it's something like photosynthesis, I also do not know what plant they use. I also don't know what chemical they use, right? So you have to pandai lah. If you can answer this, that means you're very good. You really know your stuff, okay? And you know the technique untuk menjawab, okay? Which I'm trying to impart or tell you now. All right, okay. So that's my class for today. So in a nutshell, that means summary. Remember your three main ideas. Start your question is, what is X? Okay, X is diffusion is, photosynthesis is, transpiration is, and then you use everything that is given there, masuk. You use what plant, use what chemical, use what solution, masuk dalam. Okay. And second point, how you measure it, okay, is determined by, is shown by the RV. Third point, this X is affected by MV. All right, okay. So I hope that you have learned how to answer defining operationally and won't have any more doubts how to answer it. Okay, if you still do have doubts, no problem. You can contact me. All right, you want to give me any experiment that I can try to, give you an answer or try to answer to the best of my knowledge as a teacher lah. Okay, so I'm going to give you my Telegram name. All right, if you want to contact me, very good. Okay, I don't mind. Right, so I'm going to copy it here and okay, here. Wait. Any questions? Yeah, correct. Definition. What is? What are you asking? Ask you what? Apa yang dia tanya tu? And then second point, RV. How do you measure that thing there? Shown by what? Measured by what? Determined by what? Third point. This thing X is affected, influenced by your MV. Yeah, the factor, the MV lah, not just the factor, Atira, it should be MV lah. Okay, so this is my Telegram. Okay, Telegram. All right, so you want to contact me, just go to Chong PY, and my YouTube channel is here. You can see more videos that I have shared lah. Actually, I started doing videos like 4 or 5 years ago, right? When I started NCO lah, right? So and I find that I very I get motivation from what you call feedback from students. They say it really helped them a lot, and that gives me motivation to do even more. Okay, so all so that you sometimes not everybody gets chance to a good teacher who explains everything well. So depends on your luck. Sometimes you get good teachers, sometimes you get teachers who can't really tell you what you really want to know. So maybe this is a time for you to find other sources, right, to learn more on your own. So if you do that, that means you are a very good student. Okay, so I hope to see you later. Of course, next week is your Amani Science. If I can, I'll make some more videos on the paper 2, right? Because after paper 2, you don't want to see anymore. Paper 3, you see paper 3. All right, thank you. Yes, Atira, so much. Thank you everyone for joining. Thank you very much for your comments, and also you ask very important questions. I'm sure other students also want to know. All right, you ask good questions, and by asking, you actually learn more. Okay, all right, so I will see you another time. I'm not sure yet, maybe next week or maybe the week after that. Okay, bye-bye everyone, and good luck for your exam next week. Get your practical exam. All right, okay. Bye-bye everyone. See you.